Well, as usual, it seems like turning everything off and back on again is the solution. Thanks very much for the invitation to speak with you. I'm sorry I can't be there in person, but these are the times we live in, I guess, unfortunately. So my lab is at the University of Wisconsin-Madison in the genetics department, and we're a relatively new group. And I'll tell you a little bit about what we've been working on lately. So we're interested in the causes and consequences of variation in the mutation process. And by that I mean variation in how mutations are occurring both among different genotypes in a population, as well as across a genome at different sites in the genome. And we want to do this for a few reasons. We can use these kinds of data to be able to better predict what kinds of molecular genetic variation patterns we're going to see, and also potentially to predict what kind of phenotypes will arise from mutations. And part of the reason this is interesting to me is that kinds of phenotypes and molecular variation that are caused by a mutation may wind up, in some cases, feeding back into the evolution of the mutation process itself. And so, as you probably are aware, a lot of studies of mutation use budding yeast. So this is because it's a great system for doing mutation research, grows quickly and easily. And you may know also that yeast can grow in both a haploid and a diploid form. And I was intrigued by evidence that haploids and diploids seem to evolve in somewhat different ways when people do experimental evolution studies with them. And so we wanted to understand better how haploids and diploids were actually generating spontaneous mutations, where our null hypothesis would be that diploids are simply doubled haploids when it comes to the mutation process. And so in order to study new mutations, we do what's called a mutation accumulation experiment with daily single cell bottlenecks. So we can take a single colony, streak to new single colonies every day. And in this way, the population size fluctuates over time, but the effective population size in this case will be very, very small. In this case, something around 8 individuals. And what this means is that genetic drift will really dominate the fate of new mutations rather than natural selection. And so we have 220 such lines of yeast. Some were haploid and some were diploid. And in some cases, we also have strains with the homologous repair gene RDH54 deleted. I'm not going to talk too much about that aspect of the experiment. And then naturally, after about 1,600 generations of mutation accumulation, we want to sequence these strains to find out what mutations occurred. And this is just a summary. Quite a lot of data we collected. This was published a few years ago now. We essentially find that haploids and diploids are quite different in the ways that they're mutating. And I should say that these haploid and diploid strains all were generated from the same genetic background. So the only real difference between them is their ploidy state. We see that haploids have a higher rate of single nucleotide mutation in the nuclear genome. No difference between haploids and diploids in the rate of indel mutations, although there are fewer of these to work with in the data set. And we see quite a strong increase in both single nucleotide mutations and indels in the mitochondrial genome in haploids. By contrast, the diploid strains, we saw aneuploidy, so the gain or loss of whole chromosomes, whereas we did not observe any such events in haploids. Haploids, of course, can't lose a chromosome because that would be lethal, but we know that chromosomes in some cases, and it appears that that happens at a much lower rate than it does in diploids. So haploids and diploids are experiencing a pretty different spectrum of new mutations. And I'll just focus briefly on some of these aneuploidy events where we can kind of describe the spectrum of aneuploidy, both in terms of how many chromosomes are gained versus lost. So we have 35 gains and 3 losses. That is a bit surprising. We would expect maybe an equal number of those. The rates of chromosome aneuploidy were very much unequal across the different chromosomes. And this was only weakly correlated with chromosome length. And so this is something we want to unpack further in the future, which is why do we see aneuploidy events occurring at different rates on different chromosomes? And why is it that we see so often chromosome gains rather than chromosome losses? It could be because selection is acting to remove certain aneuploidy events, and especially chromosome losses from our data set, so we don't see them accumulating. But we know from other experiments that many of those events can be tolerated when they're sort of generated on purpose in yeast strains. Another possibility I've been considering is whether there is enough time for back mutation to occur, where some of these aneuploid events are more easily reverted than others. The other pattern I want to tell you about that we found in the genome was that the haploid and diploid difference in terms of the spontaneous single nucleotide changes was not randomly distributed around the genome. And here I'm going to focus on DNA replication timing. So this is another great thing about yeast, of course, is that because so many people are studying it, we have lots of information about how their genomes work, including the timing of DNA replication. So a replication origin will fire, and that'll be an early replicating region of the genome. There are multiple origins that fire throughout the genome, but at different times. And so we can say essentially whether each part of the genome was replicated early or late. Previously, haploid yeast were found to have higher mutation rates in late replicating regions of the genome, but there was no evidence found for this in diploids. The hypothesis for why this would happen in haploids is that early on in the replication process, error-free repair mechanisms are used to repair DNA lesions, whereas later on in the replication process, translesion synthesis occurs instead, and this results in more errors in the form of mutations. So we have the opportunity now to test whether this is true in a direct comparison between haploids and diploids. And indeed, we find that replication timing is affecting the haploid diploid mutation rate. So here we see that in the earliest replicating regions of the genome, their mutation rates are very similar. But then once we look at later replicating regions, there's some divergence in mutation rate. And I should say also that the mutations that are appearing in late replicating regions also had a different substitution spectrum between haploids and diploids. So there's evidence here that the process of DNA repair in late replicating regions is what's driving a lot of this haploid-diploid difference, and this is something that we're following up on now. Of course, we want to look at some phenotypes, and the most obvious phenotype is, of course, the rate that these strains are growing. We can compare this with their ancestor and get a relative growth rate measure. It would be great to see how mutations in these genomes are related to growth rates of these strains. And the most obvious correlation way was that the aneuploidy events, those cases where chromosomes were gained or lost, had a really strong correlation with growth rates. So here I've scaled everything so that we're simply looking at genome size change. So gaining two copies of, or a copy of two different chromosomes or one larger chromosome would be scored equivalently on this kind of metric. And sure enough, the more genome you gain by mutation, the slower you grow. And so that was a very strong effect that we see, of course, only in the diploids. But later I'll talk more about kind of unpacking the relationship between genotype and phenotype here in more detail. Since we published that paper, I've been trying to look at some more traits. So these are single-celled organisms that's a little bit hard to look at them and measure traits of an organism like but we can look at cell size through flow cytometry, running lots and lots of cells through a flow cytometer. And what I find really interesting here is that those strains where genome size has changed, in general, show larger cell sizes. Genome is bigger, the cell is bigger, that sort of makes sense. But those few cases where we saw chromosome losses also had quite large cells. And so what we may be seeing here is that messing up the genome, the karyotype of these guys, causes them to grow more slowly, and they gain a larger cell size prior to dividing. In that case, we would expect to see generally a correlation between cell size and growth rate. But when we look at all of our lines together, accounting for aneuploidy in the diploids, we see a somewhat different pattern. So here at the center dotted line, the ancestral cell size and the ancestral growth rate. And so for both haploids and diploids, it seems that having either a decrease or an increase in cell size was associated with slower growth. And so what I'm thinking more about is how to unpack the, I guess, the pleiotropy, like in the talk we saw just before this one, the pleiotropy between cell size and growth rate. I would have expected that in general, slower growth would be associated with larger cell size, but instead we see that there could be some stabilizing selection on cell size, and intermediate cell size is the best. And we have plenty of mutations that seem to be causing cells to get smaller. There are some other fitness-related traits that we can study in yeast, and some are specific to certain ploidy levels. So in diploid yeast, when they will sporulate, which is a meiotic event. They form 4 haploid spores, and we can score how readily they do that. So here in the red area is where the ancestral strain of our mutation accumulation lines falls in terms of its sporulation rate. And we see that in these mutation accumulation lines in green, many lines have lower sporulation and many have higher sporulation. This is not necessarily what we would expect for a fitness-related trait. We might expect mutations to generally cause sporulation to get worse by disrupting genes that are important for sporulation. So indeed, we do see mutations in genes involved in meiosis and sporulation, things that are already annotated to have that effect down here in this category. But we also see cases like that up here where we seem to have improved sporulation, missense mutations involved in spore wall assembly. It's also clear that there's very high genetic variation here overall. Each of these strains only has perhaps a dozen mutations or so. And so it's clear that mutation can generate plenty of variation in this sexual performance trait. And this is a good reminder that even though we might knock out a gene and expect it to decrease something like sporulation ability, a missense mutation in that same gene might actually have a different effect and perhaps increase sporulation ability. Probably these strains that we're working with have not been under much selection for high sporulation in the recent past. And so we may be seeing now genetic variation that can actually restore some of that ability. On the haploid side, haploids don't sporulate, but they do mate. So the 2 mating types, A and alpha, will mate with one another by attracting one another with corresponding pheromones. And we can actually test quantitative pheromone they make using this kind of halo assay, it's called. And so here in these dots are our query strains, and there's a lawn of a tester strain whose growth is inhibited by pheromone. And so we can test all of our lines in this way, as well as the ancestors. We actually already know some have some lack completely the ability to mate, and sure enough, these form no halos. So the cause of their sterility is their lack of pheromone production. And we're seeing nice repeatable variation in halo size. Again, we would expect that mutations might generally cause pheromone production to worsen, but this was actually only true in one of the 2 mating types, the MAT alpha MA lines, which is also where we find 2 sterile lines that I'm not showing here. Whereas in the MAT A, the other mating type, we see a more symmetric ancestral value. So it may be that the genetic background that we're studying here is actually more optimized as a MAT alpha for whatever reason. And so when we add mutations, it makes them worse at being that mating type. We also see that halo size, or the production of pheromones, is negatively correlated with growth rate. And so here we have a possible trade-off between the asexual form of growth and then the ability to grow, to perform sexually and attract a mating partner in these strains. And so this is consistent with some other evidence that's coming out where the 2 mating types that produce 2 very different pheromone compounds can actually be quite different. Although they're isogamous, they're not males and females, they may in fact have lots of differences that we can still explore. and the potential for antagonistic selection between the two. So we want to try to do a better job of predicting the fitness or growth rates of our strains, given that we know what's going on in the genome of each of these strains. And so yeast experiments should give us a good opportunity to test methods like that. We have replicated fitness measures in a common environment. We have a set of strains that only differ because of a handful of new mutations, so they're on a common genetic background. And our sequencing of these strains is pretty effective. We should have a near complete picture of all the genetic variants in each strain. And so given that we have essentially a clear picture of the genotype, can we predict phenotype? In particular, can we predict fitness? We know already that aneuploidy has a strong effect on diploids, so we're going to account for that. We may be interested in whether mutations are occurring in genes that are considered essential, so genes in which a knockout would be lethal. And we can also consider methods, non-synonymous mutations in protein coding genes as either deleterious or not deleterious based on sequence conservation. So that's what I'm going to talk about next. We use this method called Provian, and it has been used to predict whether variants are deleterious in quite a few different organisms now. It's really, really popular in ecology and evolution studies. As you can imagine, if you're working particularly in a field-based organism or something where experiments are really challenging, you don't have very many direct ways of testing the effect of a mutation, and so tools like this could be really valuable. So it's been used in viruses, bacteria, crop plants, trees, fungi, mammals, birds, fish, and insects. So it runs the whole gamut of studies. And so what we wanted to do is apply the Provian method to the non-synonymous mutations, including the indels, in the mutation accumulation lines that I've been talking about, as well as some other data sets where we have both the genotype and measures available. And so that's both the haploid and diploid yeast that I've described already, diploid yeast from another mutation accumulation experiment, and also haploid green algae from a different lab entirely. How does Provian work? In brief, you are submitting a query protein plus a variant, and then that protein is blasted against all of the available sequences. After some clustering, you get alignment scores that might differ between your sort of ancestral protein and your mutant protein. And the difference in those alignment scores is what's referred to as a Provian score, and may tell you the extent to which this variant might be underrepresented in evolutionary sequence space, and therefore more likely to be deleterious. And so what we're doing is all the variants in our lines, where we know growth rate, and we're going to see whether these Provian predictions are a good predictor of fitness, or whether some other kind of predictive approaches actually work better. And so the default cutoff for Provian is -2.5. So anything smaller than that is considered deleterious. And that cutoff score, it's important to recognize that it's based on human disease variants. This was designed to increase the power to detect human disease variants. I'll show you just some of the results from this study. So this is our diploid yeast, and we're accounting for aneuploidy effects here. So this is a partial regression. And so what we can do is we can say, what is relative fitness as compared to the number of mutant proteins? And we get a relationship here where if there are more mutant proteins in a strain, fitness is lower. If we do the same thing with only the number of proteins where a variant was scored as deleterious by Provian, we get a very similar fit, but it's actually a little worse. Yes, you have a worse AIC score here. And so what this means is that actually including all of the non-synonymous variants in the data set actually gave a significantly better prediction than including only those variants that were scored as deleterious. And that was somewhat surprising to us, given the popularity of this method and the sort of high level of control that we have over our genotype and phenotype measurements. There was one exception, which is that in the algae data set, we found that the sum of all Provian scores was the best predictor of fitness. So not the number of variant scored as deleterious, but actually summing up the raw scores for some reason led to the best fit. And when we chose to look at only a, that always gave a worse model fit. So that wasn't particularly helpful in this case. I mentioned that the default cutoff score is negative 2.5, and one way we can look at these data is by adjusting where we put that cutoff score. And so here, these plot in gray the proportion of variance included, going up to 1 here, and then the AIC score for the model fit to the fitness data, where the dashed line is the default score. And as you can see in these top 3 panels, when we're including more and more variance, we get a lower and lower AIC score, which is a better model fit. In this case with algae, we see that the cutoff of about negative 0.2 would be best. And so again, this is recapitulating what I already said, which is including as many variants as you can actually seems to provide the best prediction of fitness. So that's our result, and we should think about what that means for all the studies that are using this approach. I would have thought that genetic and environmental controls would make it easier for us to detect correlations between these scores and fitness. But we should also recognize that the amount of evolutionary variation out there in the world that we see that protein makes use of, that may come from the average effect of some variant across many genetic backgrounds and many environments over many generations, whereas what we're measuring here is its effect in a given genetic background or a given environment. I still think this implies that we should use caution when applying these predictive tools outside of the context in which they were calibrated, and it may be that the best conclusion is that what protein is detecting are things that are detrimental in the long run on average, but that might not mean that they're detrimental in the particular case you're studying. I'm going to turn now to something else that we've been working on with these yeast MA lines, which is looking at something that we didn't initially have time to cover in our first publication. So this is mutations in repetitive DNA. Repetitive DNA is really hard to study, but we know it can be important in many cases. And so what I'll talk about here is, in particular, the ribosomal DNA locus. So ribosomal DNA encodes ribosomal RNA, and ribosomal RNAs, along with some proteins, together form ribosomes, which synthesize proteins by translating mRNA. So these are pretty important molecules. And each yeast cell needs, let's say, 100,000 or so ribosomes. So there must be lots and lots of transcription of ribosomal DNA. And accordingly, these genomes have many, many copies of the ribosomal DNA sequences on chromosome 12. So there's just one array of all of these tandem repeats. Each chromosome 12 has somewhere in the neighborhood of 50 to 150 copies, it's believed. And each of those copies is about 9 kb long and encodes a bunch of different ribosomal RNAs. Okay, so having lots of copies of this particular sequence is good for making lots of ribosomes and making lots of protein, but there's a problem, which is that having repetitive DNA in your genome can potentially be unstable. The repetitive nature of this region means that copies can be more easily gained or lost because of errors in replication and recombination. And that's particularly true in this region where we not only have repetitive DNA, but highly transcribed repetitive DNA. So one way that things can go wrong is through intrachromative recombination, where you lose a whole circle of copies and you get copy number loss. And so if ribosomal DNA copy number, if it's advantageous to have a stable copy number, we might expect that evolution has created canalizing or homeostatic mechanisms. And that does indeed appear to be the case. So this is not my research, but kind of a summary of lots of other studies. Essentially, what's happening here in this model is that when total copy number is low, there is a regulatory mechanism that increases the likelihood of unequal recombination between sister chromatids during DNA replication. So in other words, you've got a replication bubble, you've replicated out some copies already, but a double-strand break occurs, and the nascent sister copy is then used as a template for to copy onto this other strand, leading to an increased copy number on the leading strand. And so in this way, it's thought that there's a homeomorphism where copies are gained when copy number is low. And so what we wanted to know is whether this is actually happening in our mutation accumulation lines. What happens to this copy number if natural selection isn't acting strongly? Do mutations throughout the genome break down the mechanisms that maintain this repetitive locus? And then what are the fitness consequences of that variation, as well as the mutation spectrum within the ribosomal DNA locus itself? There's a couple of methods that we could use to assess copy number. We do already have sequence data, so we could look at sequencing coverage. This is the kind of thing that you see if you do that. The reference genome doesn't have 100 copies of this locus in it. It only has 2. And so what you'll see is a really big spike in coverage right at that location. And in principle, the size of that spike should reflect the copy number, how many copies actually underlie that repetitive region. This is great because we already have all the sequence data, we can look at that, but it's been unclear for researchers how exactly to use this coverage spike to calculate copy number. And so we did want a separate method as well, and so we used digital droplet PCR. In this method, you cut DNA using restriction enzymes, and then essentially do PCR in each droplet separately. And then the presence or absence of your template will show up. You can fit a Poisson distribution and find the average copies per droplet. So we do this for both the ribosomal DNA locus and for a single copy gene on the same chromosome as a control. And so we've combined these two methods, and here's sort of what short read sequencing might look like across this locus. Here are the 2 copies of the locus in the reference genome. These are our cut sites. These are the regions we use for ddPCR. We've got measurements, not from all the lines, but from many of them. And then what I did was calibrated the sequence-based estimates to the ddPCR-based estimates. So I used optimization to determine which regions of coverage we should look at in order to get a similar estimate to the ddPCR values. And so that turns out to be these blue regions here. It's not the whole locus. And we get a pretty good fit between our 2 methods. So what did we find? The copy number actually mostly declined under mutation accumulation. Some lines got really high, but for the most part, the average decreases. The haploids and diploids don't differ. So here I'm referring to copy number per chromosome 12. Diploids have twice as many, and so we can just imagine they have twice as many total copies per cell as haploids do. We get lots of. Just briefly, I wanted to test whether this was true in that other set of MA lines from a different lab. In their standard YPD environment, that's also what we see in those strains, as well as lots of variation across environments in the pattern of change. The relationship between copy number and growth rate suggests that there could be stabilizing selection on this locus, so at least in diploids, with intermediate values being favored, and higher low copy number being disfavored by selection. We can calculate the strength of stabilizing selection in this region and conclude that it is fairly weak and perhaps non-existent in haploids. One thing that's really fun is that there have been recent studies of natural strains for this trait, how many copies of RDNA there are. And so that's what's shown here in green. You can see we kind of have a similar shape among our MA lines. And we can actually ask whether the standing variance in this trait could be driven by mutational variants or whether we need to invoke some kind of diversifying selection. So first we can say there's too little variation here for this trait to be selectively neutral based on the mutational variance, so that's not too surprising at all. And then we can also say that the variation that we see coming from mutation combined with the stabilizing selection on this trait does seem to be able to explain the standing genetic variation. So in other words, mutation stabilizing selection balance is probably a good explanation for the variation that we're seeing in nature. We can look at some interesting patterns of molecular variation in copy number. I mentioned that we have some aneuploid strains, and that includes strains with an extra copy of chromosome 12. Those strains with an extra copy of chromosome 12 have significantly lower copy number per chromosome. The most likely explanation, I think, for this, with an additional copy that appears through an aneuploidy event, there's a higher copy number per cell, and so copy number expansion is suppressed, copies continue to be lost, and you wind up with fewer copies per chromosome. But we can't reject the alternative causality here. We see some outlier strains that we can study further. So this very high haploid line has a mutation in RIF1. RIF1 is a gene that controls genome integrity by inhibiting RDNA replication. So this makes sense that a mutation in this gene could cause high copy number. But interestingly, 2 other strains with typical copy number also have mutations, non-synonymous mutations in RIF1. And so to unpack this a little more, we actually had a look at where these mutations occur in the protein itself. So this is a homodimer that hangs on to double-stranded DNA. And here, we're pointing to this K1212 mutation. That's probably occurring in an area that has contact with the DNA, whereas these other 2 mutations would not appear to affect contact with DNA. So it's plausible that we have a difference here driven by the molecular structure. Similarly, we see that this position 1212 is conserved among several yeast species, whereas these other 2 positions show less conservation of the amino acid here. Again, this would be consistent with this position being more important for function than these 2 other positions. I'm going to skip this. We see some similar patterns in diploids. We can also look at relationships between copy number and gene ontology categories, and we can find some hits this way. This is a more statistically agnostic approach. We find that MA lines with more non-synonymous mutations in genes in the regulation of cell cycle category have higher copy numbers. So there's some link here between cell cycle control and ribosomal DNA transcription and maintenance. Finally, I do also want to look at point mutations within the rDNA locus itself. And so this is another thing that we weren't able to do before, is actually score mutations in this relatively large region of the genome, point mutations. So we identified segregating variants in this region using VarScan, found a good number of substitutions and a few indels. And if we try to calculate a mutation rate per site, per base pair here, we would see a pretty similar rate to the rest of the genome. So I think these results make sense. However, we do see quite a different spectrum of substitutions. Particularly AT to GC mutations seem to be more likely in this region than in the rest of the genome. This could refer in this repetitive region. The point mutations, excuse me, the point mutations within this locus could occur in any of the copies that are in there, right? And we can't tell from our data which copy they're in. But we can use their frequency and the total copy number in each strain to infer how many copies are affected. And we might imagine that a mutant copy could appear somewhere in the array, and that copy gain and loss, you could lose the mutant entirely, or eventually gain more copies of the mutation. And I think we see what you would expect from this kind of process. So most of the time, only a few copies are affected, 1 or 2 in most cases. But occasionally, this new mutation can actually wind up spreading across many, many copies in the genome. Within this locus itself, we can also see some pattern mutation. And so here's another kind of view of this region. So this NTS1 is repeated on either side, so that's kind of a break point between these copies. And these are the non-transcribed spacer regions. Those regions are believed to evolve quickly due to a lack of purifying selection. But actually, we see that they have higher rates of mutation as well. And that may be because these are the hotspots for DNA breaks and recombination in this array. To gain more evidence for this, we looked at polymorphism from wild strains across these regions. And our mutation data actually are significantly associated with polymorphism data. So we're seeing more mutation where we also find more polymorphism. consistent with the idea that mutation rather than selection might be driving a lot of the polymorphism pattern here. Okay, so this locus is particularly in terms of its copy number, but there is some stabilization and control of copy number, but that can go awry when new mutations appear in the genome. There appears to be a relatively large target size for mutations that can cause disruption in those homeostatic mechanisms. When those mechanisms break down, we see copy number usually drops, probably reflecting the kind of natural losses without an ability to regain copies. But we also see certain specific mutants that can get very high copy numbers, and that's probably due to hyperactivation of those homeostatic mechanisms. We can pick up some specific and general patterns of individual mutations that lead to copy number changes. And within the locus itself, we see some unique patterns of mutation that actually do match polymorphism. So we need to think about mutation in this context and not just selection. More about this work now on bioRxiv. And so I hope I've convinced you that yeast experiments, particularly mutation accumulation experiments, should be providing us many more opportunities to explore different kinds of dimensions of mutation and to test some of our ideas about genotype connections. Thanks to all my co-authors on these different projects, and thank you for listening.
Thank you very much. We have time for questions.
Thanks for the interesting talk. I just have a quick question regarding the methodology when you're detecting the copy number variations. If I understood you correctly, you use the ddPCR as well as the Illumina sequencing to detect the copy numbers. I was wondering whether you consider to use the nanopore platform to try that.
Yeah, so that would be useful if we could get very, very, very long reads. I'm not sure how long they're getting these days, but so each copy is about 9 kilobases, and there might be 100 of them. So 900 kilobases long for one array. I think it would still be challenging with long reads, but certainly, in the future, that's the type of method that should be hopefully applicable to questions like this. Particularly if we can multiplex to get through a lot of our lines at once.
It might be doable these days, just a little bit pushing the boundaries.
You're right.
Other questions? Okay, actually, I have a quick curiosity. So, what is the some details about the first part of the talk, the technique that you use actually to merge the different prediction when you use Provean? Did you use any particular strategy?
We followed the. Hopefully, this is what you're asking. We followed the recommended protocol for applying Provean. Is that?
Okay, so the protocol, like I don't know, if you have multiple mutation, are you using, I don't know, the average, the maximum value?
Yeah,
there is any specific protocol for this?
I see what you mean. So if there are multiple mutations in one protein,
yeah,
so that can happen occasionally, particularly in larger genes. We would simultaneously apply both mutations and then run Provean on that double mutant protein. This can happen particularly because we can see some multiple mutation events or complex mutations that affect multiple parts of the sequence. And then the other way to maybe think about this is, what if a strain has mutations in multiple proteins across the genome, which they mostly do. In that case, we tried simply summing up how many proteins are hit, but also summing the scores themselves for all the proteins that were hit. Hopefully that clarifies a bit what our approach was.
Okay, so just one last consideration. I think that maybe the data that you are generating, since here there are the organizer KG, maybe this experimental data can be turned into a challenge of cages. So maybe some of the organizer can contact you and see if something can be organized. So Pedja is here, and I think maybe it's interesting.